Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Mark Erlander. He's the CEO of Cardiff Oncology, and we're going to talk about his work with colorectal cancer and what he's discovered. So, Mark, thanks for coming.
Thank you for inviting me to your Finding Genius podcast. I'm honored to be one of your podcasts. Thanks for your interest in Cardiff Oncology.
Yeah, tell me, how did you get interested in cancer? How did you cross paths with it?
Well, it was about 20 years ago, or a little more than that, actually. I got started looking at cancer way back in when I was working for Johnson & Johnson as a research fellow. We were looking at a lot of databases of, sort of the genetics and the genomics of tumors, because that was really exploding during that time in the mid-1990s and 2000s. So that's how I got started, and I got involved in diagnostics in oncology, developed some tests, breast cancer and metastatic disease, and then got involved in this actually at Cardiff 4 years ago. But I've been very, very interested in oncology simply because, when I was looking at things to do as a scientist, it became really clear to me that with the whole genome being sequenced back in over 20 years ago, this is really the first place where you could really use that information was clear to me was in oncology. And so I wanted to do something and try to make a difference in my lifetime on something. So that's how I really got started.
Okay. And then of the cancers, why colorectal? Why the focus there?
Well, at Cardiff, we're in multiple different indications. We're in prostate cancer, we're in leukemias, but our flagship program is in colorectal cancer because of what we're seeing from an efficacy point of view and the fact that I do believe in colorectal, we have two shots on goal because the tumors, 50% of the metastatic colorectal cancer tumors have this really deleterious oncogene called KRAS. PLK inhibitors such as ours, Onvansertib, can really kill those tumors more readily than if the tumor type did not have the KRAS mutation. And then on top of that, the standard of care today that they use for these patients is this chemotherapy. It's a cocktail. And we actually have, with our small molecule oral drug, we have what they call synergy with these chemo drugs. So these chemo drugs are given to people, the standard of care. And we found in basically preclinical models that we have what we call a synergy. That is to say, unexpected result in the sense that 1 + 1 = 3 with regards to killing tumor cells. So the pathways sort of interlace with each other and cause a greater effect than one would expect. So because of those two things, we got colorectal. We really went after it aggressively.
So colorectal cancer, what's the tropism for the metastatic sites? Where do they tend to go?
Well, 50% of them go to the liver, but also peritoneum, and lung, but most, a lot of it goes to the liver, but also to some other areas such as lung, the sort of your gut.
Okay. I know tumors are heterogeneous. So is your inhibitor, I guess it's an immunotherapy type action, and is it target specific metastases or anywhere there's tumor burden?
Well, yeah, that's a great question, Richard. So Onvansertib, which is the name of the drug, first thing you have to learn when you get involved in drug development is that the names that they have you come up with are all very hard to pronounce. But anyway, so Onvansertib is our drug, and it actually inhibits an enzyme. So it's not immunotherapy, but it's actually, there is a key enzyme that allows tumor cells to go from one tumor cell to two tumor cells. In other words, the cell division. And tumor cells are much more vulnerable to the inhibition of this enzyme than normal cells. So you have a therapeutic window where you can preferentially kill tumor cells by giving Onvansertib, our drug, because it really goes after a very specific enzyme that's critical for tumor cells to divide.
Well, chemo, from what I understand, it goes after fast dividing cells. But with your drug, with this missing enzyme or with this enzyme, wouldn't that slow down proliferation, and then chemo would be less effective against cancers?
Well, what happens, it's a great question. So what happens with some of the chemos given, and particularly the ones in colorectal, which are named irinotecan and 5-FU, are the two chemos that are given in combination. What they actually do is they cause DNA damage. They cause the tumor cells, the DNA of the tumor cells, to be damaged. So when that tumor cell is starting to divide and go from 1 to 2, there's a natural process or a checkpoint that all cells have, normal cells, tumor cells, which is that they try to repair the DNA. Now, in the case of if you give chemo, what happens is the DNA damage is so great that usually that would result in the tumor cell going to cell death. We just can't repair the DNA enough, so it just dies. But in tumor cells, they overexpress and hijack this enzyme that we target called PLK1 is the enzyme. And so PLK1 actually suppresses that stopping and allows them to shoot right through to cell division. So where chemo can fall down in the sense of not be effective is when the tumor cell can divide, even irrespective of having DNA damage. But what they do is they hijack PLK1. And PLK1 is a very efficient enzyme, very pleiotropic enzyme that controls cell division. And it suppresses that checkpoint of DNA damage. And so tumor cells can ride right through in without dying because of the DNA damage. So they actually work well together, and that's part of the reason for the synergy. You DNA damage and inhibit the PLK1.
So tumor cells recruit PLK1, I guess, in abundance so that they can continue to divide?
Yeah, PLK1 is overexpressed in tumor cells. And they're more vulnerable to inhibition of PLK1 because of that. They rely on it. They're very dependent on PLK1. So it's kind of a little bit of one way that can think of it is it's PLK1 is an Achilles heel for tumors, because if you can inhibit that, you can stop those tumors from dividing.
All right. So the cells produce it themselves, or do they pull it from the interstitium, or where does it come from?
Tumor cells, all cells have PLK1. It's an enzyme. It's involved in the cell division process, but tumor cells overexpress PLK1 and are more dependent upon it and more vulnerable and hypersensitive to the inhibition of that enzyme. And so you can kill a tumor cell, and at that same concentration, a normal cell will be unaffected. So that's really the premise. That's the crux of the argument for targeting this enzyme in tumors in general.
So how do you administer this?
It's an oral drug, and so it's a small molecule, and it's oral, and it has a half-life of about 1 day in humans. So it actually goes away relatively quickly, which is also a good property.
So how far are you in terms of clinical trials? What model are you using? Organoids or animals?
No, we're in patients. And so we have 3 ongoing clinical trials right now. And one is in leukemia. Another one is in prostate cancer, specifically castrate-resistant prostate cancer. And a third is the one we're talking about, which is in metastatic colorectal cancer, specifically the KRAS mutated form of the metastatic disease, which, like I said, is 50% of all metastatic colorectal cancer patients have that. But we also just got the May Proceed letter from the FDA to start a trial on pancreatic cancer. And so hopefully we'll have a patient dose before the first half of this year. But that we're very interested in pancreatic because, we've been talking a little bit about this deleterious oncogene called KRAS. And I said it was 50% in metastatic. Well, in pancreatic cancer, it's 95%. It's pretty much all patients.
That's good.
Right. Pretty much all patients who have pancreatic cancer, it's driven by KRAS. And so we believe that we have a shot at it, given the data that we have so far in colorectal cancer. So it's and that, of course, is another, I think everybody on the planet knows that that's a huge unmet need is pancreatic cancer.
What's the effect when you administer this? What happens?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
It's actually well tolerated. In general, I would say that we have not, at the doses that we give the patient based on our data, they really don't have any GI problems. They don't lose their hair, no alopecia, and there's no myelosuppression. That is to say, white blood cells down or neutrophils down at the doses that we give. So I would say, in overall, it's a well-tolerated drug.
But what's observed with the tumors? Do they shrink? Do they just simply stop growing and allows chemo to attack them? Like, what happens?
Right. So all of the above. So what we see in colorectal cancer is that depending on the patient, some patients have significant tumor shrinkage. Over 50% of the tumor shrinks, and it's enough for them to then go on to a surgery. But also, we do have patients that it's really what we call stable disease. That is to say, we stop the tumors from growing, but we don't shrink them that much. So there's obviously the question would be, well, why this? And I think you already mentioned the word heterogeneous, which is was issue, and it's a problem. Is that even within a given tumor type, like what we're talking about, colorectal cancer, the underlying drivers and the underlying pathways that drive that tumor to multiply and divide can be different in each patient to a certain extent. So sometimes you get much greater responses and others you don't. But it's usually, and the reason for it is the underlying genomics or underlying genetics of that tumor.
So when would this be used? Is it with the first set of chemo? Is it, if someone's in remission, would you pulse it to them periodically?
Yeah, I mean, right now, this drug has not been approved by the FDA yet, and so we are not going into a first line situation. We do have to do a bit of proving ground and showing the efficacy and the value of Onvansertib. So we are in metastatic disease clearly, but within that we're going after what is called second line therapy in metastatic colorectal cancer. So what does that mean? Well, that means that the patient, in this case, these patients, the half of the patients that have the KRAS mutation, they first get a chemo cocktail. It's called FOLFOX. It's an oxyplatin-based chemotherapy. And then once they, for those patients that progress after a certain or relapse after a certain amount of time from that, then they are eligible for they become second line. And there they get a different chemo backbone, which is an irinotecan plus 5-FU. And that's where we would play a role. Because when you look at the response rates in colorectal cancer, for first line, patients do reasonably well. I mean, 50% of the patients will have tumor shrinkage. But it's only after those patients that then relapse after that first line treatment that the numbers, the prognosis is really poor. 10% survival in 5 years, and response rates are a single digit, 5-10%. So you really have this precipitous cliff in second line, and this is really where it desperately needed to have another option for these patients to extend their lives. So
when does KRAS tend to show up? I know it depends on the cancer, but does it show up in primary tumors, or is it much higher expressed in metastases?
It's a driver. Yeah, great question. It's a driver for cancers in colorectal cancer. It does drive. It's an earlier event. It's one of the early events of the tumor in colorectal cancer. So it's something that occurs relatively quickly as the tumor starts to develop. So it is considered a driver, tumor genesis in CRC, and the same in pancreatic cancer.
So what happens with, does the efficacy fall off as the tumor expresses differently to get around it? Or is this such a fundamental expression mechanism for cancer that, this therapy works, and because, again, the cancer just cannot adopt around it?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Unfortunately, tumors have ways of getting around, therapies. So we don't have 100% response rate in our trials. So we're, right now we're reporting about 35 to 40% versus standard of care, which is 5 to 10. So we're clearly way above what's out there today for these patients as we sit here today with the data. It's early data though. But I'd say that, yes, in general, tumors, it's a whack-a-mole kind of concept where you pop one thing out and then they pops up somewhere else. And so, we continue to, with us here, what we're trying to do with Onvansertib is we're adding it to, like, for instance, chemotherapies in this case, and anti-androgen therapies in prostate cancer, where in a non-chemo combination there, we really are adding, blocking yet another pathway so that the tumors have a less likelihood to find a resistance mechanism or resistant pathway. But it's a constant.
Well, what's been observed when they do find a way to get around, this enzyme, what do they do instead? Can you determine that?
Well, we at this point in time, we have not gotten down to it's a great question, gotten down to the molecular underpinnings of the resistance. That's something that we are studying. I'd say that from a clinical point of view, of course, the patient's tumor starts growing, which is obvious. But that's what you see, is that the tumor starts to grow again. But the question I think you're asking is, well, why? And we don't know exactly how that is now yet, but we are studying that in preclinical and also in patients. We're looking at we look at the entire genetic profile of each tumor that a patient that's on the trial, and to look for clues and for correlatives of those that progressed versus those that had tumor shrinkage. So we're trying to draw those investigative lines, but we'll know a lot more as we get more patients on this trial.
Are you looking into experimentation to figure out the mechanism by which the cancer is evading, yes, this enzyme restriction?
Absolutely. We're really two roads there. Road number one is one that I was just talking about, which is the sort of what we call a genetic profile of the tumor. Now, when I use the word genetic, some people say, "Oh, you're talking about, germline." No, I'm talking about the DNA, of that tumor, and what is the makeup of that DNA? Does it have mutations? Which mutations are they? And does the mutations that patients that progress, and do they have certain mutations that those that have shrinkage don't have? So are there obvious differences really at the clinical level where you're studying the molecular makeup of each patient's tumor? The second way that we're doing it is more experimental, in the sense that we're looking to see in tumor cells that you can grow in culture in the lab, whether or not, what is the resistance mode? When you add our drug Onvansertib onto those cells and you continue to do that, you'll kill a lot of them, but there will be, some that survive. And how did they survive? Now, the big question there is that you will find mechanisms of survival of tumor cells in a petri dish, but whether they equate to what's going on in patients, of course, can be a Grand Canyon of leap. But, those are the two ways that we look at it.
Well, what about using like an organoid model? I don't know if there's colorectal organoids, and perhaps that would be a little closer, or is mouse model close?
No, it's a great point. We are talking to doing a collaboration with organoid in CRC. And so that's another area that we will pursue. We don't have the expertise here at Cardiff, but we can collaborate with them. There's several academic groups that have them up and running. So there, what you could do is take organoids that they have already in freezer that they grow, and then you can look for resistant mechanism with Onvansertib. We simply, we got in and wanted to see if we had efficacy. Now that we're seeing efficacy, now obviously there's reasons to spend money and resources to look at resistance mechanisms.
Well, I know no one wants it to happen, but if your life is at stake, is there one of the cancers that you work on where a biopsy is not horribly invasive? So you could give this to them and do a biopsy, a month or so down the road and see, what's the change been?
Yeah, I mean, that's, of course, and that of course is the ultimate thing to do, to your point. It's hard to do that within those are usually very difficult to implement. There are some academic groups that do that for different cancer types. Like, for instance, we're hooking up with a academic group out of the Oregon State Health Science Center, where they're doing a trial where our drug is going to go into patients, and we're going to get a biopsy 10 days after 10 days of daily treatment, or they've taken our drug daily for 10 days, then get another biopsy and see what the difference is. So those kinds of changes and looking at those are the ultimate ways to do it.
Well, where does this KRAS mutation show up? Does it, what if it shows up in a melanoma or something that's so surface that a biopsy would be not trivial, but very doable? And then maybe you could see it.
The majority of KRAS cancers, I mean, they're about, across all cancer types, I think about 25 to 30% of cancers in general have a KRAS mutation, but the majority of them are in pancreatic, 95% of them, colorectal, 50%, and then in lung cancer, non-small cell lung cancer, I believe it's in the 35% level. Those are your major cancer types. Melanoma is I'm not sure. There they have BRAF mutations, which is a different kind of kettle of fish, but it's in the same pathway as KRAS. But I think, the answer to your question is the one thing that we are thinking about doing that we can do in the pancreatic trial that we're started, and we actually are doing this, is we are doing organoids in pancreatic, and then looking for sensitivity to Onvansertib within the organoid of the same patient that's being treated, and being able to look at the underlying genetics of that tumor. So there we might have, I think, because these organoids we can set them up, and we're working with a group out of UCSD here down the street. And so we're excited about that. There we might be able to get some clues on the resistance mechanisms.
Are you able to maybe fluorescently tag this enzyme and see where it goes in the cells and how it's used and how it travels throughout the body?
Yeah, I mean, it's the PLK1, the enzyme is quite well characterized in the sense of what it does in the what we call the mitotic machinery, that is during the cell division. It has some multiple functions in that last part of when cells are going from 1 to 2. And so, there's been a lot of basic research that's been done on PLK1 and how it interacts with a whole host of other proteins, enzymes that are involved in what we call the mitosis or mitotic machinery of a cell dividing. I would imagine that people have fluorescently tagged it and looked at it. PLK1 was first discovered out of a yeast, studies in yeast, and so it's well known the function of PLK1. But the question really came down to is how good of a drug target is it? How do you inhibit it and get efficacy?
Yeah, well, I just wondered if you're able to tag it, then in any model that you run it in, you could get more insight as to actually what's going on, because you said like in vitro is obviously very different. Cells sitting in serum versus like in situ in the body is maybe totally different with signaling. So maybe we can't without being invasive.
Yeah, we can't really do. It's an internal enzyme within the cell, and it can't be readily fluorescently tagged in patients, if that's what you're asking. It can't do that, but we can look downstream of it in patients to see what it's hitting and what it's phosphorylating and what it's doing. But it's harder in patients to kind of dissect that process.
Okay, just wondering, trying to help you, give you some ideas.
No, I'm all ears. You never know when you get the good ideas, and so I'm all ears. But yeah, I mean, we do have some, to your point, what we've been talking about. I mean, it would be great to have the ultimate pre and post tissue biopsy.
What do you think is possible in the next year or two? Short term. We're almost done, by the way. Short term, next year or two. What do you think you're going to progress to and figure out by then?
Well, I mean, our focus is pretty simple. We've got to do all we can to get this drug across the threshold and get it to at least an accelerated approval or a conditional approval so that we can get this into patients. So, our job as the drug developers and scientists here is to really push as hard as we can and be able to do the clinical trials with the rigor that the FDA will give the nod to and we will get this into patients. So in the next couple of years, that's what we're doing. I mean, that's what it takes that kind of time. So that's for us. I think it's the most important thing is to stick to as they say, stick to your knitting, and just keep your head down and focus and execute. And that's what we're doing. And our company, Cardiff has got the funds, has the runway to be able to do that. So we're well funded. And so really we have no excuses. We have to focus on what we're doing to get this across the threshold.
Okay. Well, very good. What's the best way for people to keep tabs on progress and to find out more about Cardiff?
Sure. I mean, we're publicly traded. CRDF is our ticker, NASDAQ. You can go to our website, www.cardiffoncology, all one word, .com. We have our website is very user-friendly and tells you all the latest and greatest of Cardiff and what we're doing and what we've presented and press releases and posters and presentations. So it's all there. And so that's one way to keep up. If you're really, really interested, you can contact us to points so you get on our quote unquote mailing list where you get information emailed to you. So.
All right. Well, very good. Well, Mark, thanks for coming on the podcast. I appreciate it. And
Richard,
what you're doing is critical. So thank you.
Thank you so much for your interest and in talking to me and giving me the opportunity to talk a little bit about Cardiff Oncology and what we're doing.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.